We're doing an art project involving photosynthesis, involving Arabidopsis thaliana, and involving zinc fingers. And I think most people, myself included, before I started this project, they don't know what zinc fingers are or what they can do. So how do zinc fingers work? What are they? And what might they be useful for?
Yeah, what people understand when they talk about zinc fingers is that these are small modules which are present in proteins, and usually these are DNA binding proteins. And they have a small part of about 30 amino acids, which is holding a zinc atom. And well, this structure is some kind like a finger-like structure, and this contacts DNA. And each zinc finger contacts 3 base pairs of DNA. So you can, by itself, just one zinc finger module is not that useful, because a 3 base pair sequence will occur so many times that it is too abundant. But if you stitch more zinc fingers together, this structure, this proteinaceous structure, is recognizing a longer DNA sequence. So if you stitch, for instance, 3 zinc fingers together, you will recognize 9 base pairs of DNA. So not the recognition code is not completely specific, so there is some bias, but generally you can state that each zinc finger combination will recognize its cognate DNA sequence quite well.
For me, what this means is that it's almost like a gambling machine. You have like 4 possible base pairs that make 20 amino acids, and now you have basically 3 amino acids, or 9 base pairs. And these zinc fingers are landing in the genome anywhere those happen to come up. So sort of like the lottery, you have a winning combination, but in this case, the zinc fingers can land sometimes in hundreds, sometimes in thousands of places in the genome, and set up shop there. Is that? I'm using strange layman's language, but what do you think?
Yes, it's getting closer to the idea. So once these zinc fingers were discovered in nature, their recognition code for DNA was established in the US at the Scripps Institute predominantly. And these codes were published already more than 10 years ago. And now, people started to realize that you could then specifically address sites in the genome. So, for instance, a 9 base pair sequence, which is recognized by 3 zinc fingers, is still very common. It occurs in a small plant genome, like Arabidopsis, which I work with, about 1,000 times. So each 9 base pair sequence, each specific one, if you fill down in the base, the bases, can occur roughly about 1,000 times. But if you would make a longer module involving, for instance, 6 zinc fingers, then you recognize, for instance, 18 base pairs. And an 18 base pair sequence can be unique even in a complex genome as the human genome, which is a few billion base pairs. Right. So there you could make a DNA binding module which recognizes one specific site in the genome. Right. It might interact with other sites, but this interaction with the 18 base pairs is for a 6 finger structure probably the most stable interaction that's possible.
I think about gene therapy, and I know that there's some of the uses for zinc fingers might be gene therapy, and the reason I think of that is that if we want to get extra genes into the human genome or genomes of other organisms without knocking out genes that are useful, we need to be able to specify where these new genes are going to land and insert. Mm-hmm. And with something like this, we could actually specify the placement of inserts, right? So is that one of the ways that zinc fingers is being used?
I think there are very different approaches. I think one approach could be that you knock out a certain gene, or you make mutations, and you could do that by fusing a nuclease domain, so a DNA protein domain which cuts DNA, so it breaks it apart. And if you fuse that with these zinc finger domains, you might target this nuclease to a specific site. So there it might introduce a break in the DNA, and there, at such a break, you could insert another DNA sequence. But there are also more complicated issues or strategies where they have fused zinc finger modules, so DNA binding modules, to integrases, which are proteins which are used by viruses, for instance. and they try to make these virus structures integrate at a specific site. And this is not completely successful yet, to my knowledge, but it might be another possibility to do this. Another one is that you make transcription factors. So you fuse the zinc finger domain to what you could call an effector domain, which might enhance or even repress, depending on your choice, the activity of a gene nearby the landing pad. And this strategy is also, I think, in the Scripps Institute have pursued this approach. I think also for repressing new growth, let's say, enhanced growth of blood vessels to cancer tissue. So they could repress it, I think, by having a zinc finger there, which represses the gene which was involved in widening the arteries. But I'm not a mammalian biologist, so I might not be very accurate here. But these so there are multiple things you can do with zinc fingers. You should consider them as modules which are binding DNA, and you can choose the specificity, or you can deliberately do without any adding much specificity by just going for a multitude of genes rather than single ones.
This is, I mean, one of the things you work with is this whole genome reprogramming, which actually has to do with these, say we use 9 base pairs, like 3 zinc fingers in a row, letting these zinc fingers land all over the genome and either promote or repress gene expression downstream from wherever those zinc fingers land. And so you're actually, I mean, as far as I can tell, you're actually sort of, we had talked about kicking or pushing the genome pretty hard. Instead of making one alteration, you're actually like almost seeing if you can get like mass effect, right? So you're looking, it's almost like a hypermutagenic motion towards just seeing what the plant can become under an extreme genetic stress or an extreme genomic alterity. I call it alteration, like an altered state, right? Maybe I would let you talk because I'm putting words in your mouth. I don't know if that's your research exactly.
You're very correct in summarizing what we try to do here, and if you could ask yourself a question, is that if you have a certain genome, so a complex set of DNA molecules, you might wonder what will emerge from it. If you would put this into a cell, what could it be? And well, if you have a certain species, which we recognize, it might be a plant, it might be a yeast, might be a mammal. It might still possible that having this with the same genome, it could be something else, not completely different. Maybe still recognizable as an animal, but it might be different in shape, in phenotype. So with the same genome, it's very well thinkable that you could generate an organism which is different. But that depends probably upon the interplay of all the genetic traits. And if you could kick the system that a lot of these traits are expressed at different levels, you might hit a window where the organism really starts to be different. So then you get a mutation, let's say a mutant organism, which essentially has the same genome. But it is just, like if you compare to a piano, then someone is playing another piece of music on the piano being the genome.
I know you referred to this as genomic interrogation, which I love, but I think since we're trying to actually, for public education's sake, we want to let people know, part of what you're saying is that, how do I say this? A lot of people are not aware that their genome is not just them, but actually their gene expression patterns are also part of their inherited makeup and their environmental interaction. So like I've said to you, right, I know that if you drink milk, it's only at that time something in the milk, whether it's the lactic acid, actually trips a promoter in your genome that starts you to digesting the milk product. And before that, you're not digesting it. So maybe you want to talk just in regular speak about what it is that gene expression, like up and down regulation is. In particular, if we're talking about effects of reprogramming up and down regulation patterns, I think you started on it, right? We want to talk about what it means to upregulate and downregulate your gene expression, but then also, I mean, I think we're heading into a place where we might talk about what might be in the genome that's not expressed ever, that only gets expressed when the genome is interrogated. So I guess it's two questions. What is up and down regulation, and what might be in the genome not usually expressed, but after interrogation might actually come up from deep time?
Yeah. Well, this is an intriguing question, of course. Well, if you consider one particular gene, and I think most molecular biologists will agree that you could say that if you talk about gene expression, well, at the level of RNA levels, that the level of RNA depends upon the number of transcripts which come forth from this gene per unit of time. And it also, of course, depends upon the stability of this messenger. But let's say that it is the number of initiation events. So meaning that each transcription event is an initiation of transcription. The more transcription events you have per time unit, the more highly expressed the gene is. So you could also repress this gene by knocking down this expression levels, meaning that less transcripts are generated per unit of time. And how this happens in nature is that somehow the transcriptional machinery is assembled nearby this gene, so at a position where the transcription really starts. And you could say that activators of transcription help to recruit all the factors which are necessary for gene expression to this particular position, which is critical for initiation of transcription. And repressors might just do the other thing. They recruit factors which tend to make the gene inaccessible for these factors. So this is when you look at a specific gene. So you might try to influence this. Well, you could do this by zinc finger technology probably. You can make artificial transcription factors. You can make them land in the neighborhood of this transcription initiation site. And then if you have an activator there, then you might recruit factors which help to stimulate transcription.
This would make multiple or extra copies of the proteins that come from the gene expression. Yeah. So when the gene is transcribed, it makes more proteins or more copies of protein. Yeah. Sort of like saying, how many copies do you want of this gene? Yeah. Or how many copies do you want transcribed from this gene? Or what would you say, this is a weird question, but when I say that downregulation is causing a gene depression, or upregulation is causing a gene mania, where it says, I'm going to just make 1,000 copies of this today. It's alliteration, it's a little lateral thinking, but I mean, what do you think about the concept?
Well, I think that's right. What you say. That's indeed the concept. So you could look at one, but you could look at one gene as I just did, but you could also have a bigger picture in mind. That you say, okay, I try to influence the expression of many genes at the same time and see what happens, or repress genes at multiple sites in the genome. And then you enter the area of which you also called genome interrogation. That if you start to do something which you don't really know what will happen, but you largely disturb the system by adding, in this case, a novel transcription factor, which nature has not seen yet. It's made in the lab, and you introduce this in the organism or in the cell of interest, and then you let this thing do its job. So if you have created something which is a DNA binding module with a known activator of gene expression or a known repressor, so such a factor might bind to all its cognate positions for the DNA binding moiety, which is the zinc finger domain. And these might be multiple sites. For instance, Arabidopsis, I already mentioned, there are about 1,000 sites which match with a specific 9 base pair sequence. Such a transcription factor might be present then at 1,000 sites, and if there are genes nearby, then the conditions might be met that these genes are upregulated in case of an activator, or downregulated in case it's a repressor.
I mean, I can't help mentioning, because this is just what we did an hour ago, right? Yeah. We took these sort of 16 Arabidopsis plants, and we took off all the flowers and just left the sort of, what are they the
But
the buds.
The buds, yeah.
We left the buds, and then we dipped them in this agrobacterium that has sort of a vector that will insert the zinc finger in these upwards of thousands of places in the genome. And then we mixed it with two different transcription factors, an upregulator, what was it, VD16?
VP16.
VP16.
Viral protein 16. Yeah.
Viral protein 16, and EAR, which
Here,
is,
yeah. I always forget.
it's a repressor.
Yeah, it's a repressor.
Okay, we can go with that, but
Abbreviation, but
yeah. See, I told you his laugh is what I love the best. So you're great, Bert. So like, how do I say, like, but we just did this, and we actually added a repressor and an activator at the same 9 base pair zinc finger. How often is this done in Arabidopsis work? Is it done very often?
Well, I think this is not done very often. So I think this normally I wouldn't do this really, but myself. But yeah, it's done now. In this case, you have used a zinc finger module, which we found to be associated with development of bigger plants when it was fused to a VP16 domain. So we found a mutant plant. So you could say we did a genome interrogation experiment to look for plants which would perform better, making bigger leaves in shorter time periods. And from this plant, we re-isolated the zinc finger module and reintroduced it into plants just to see whether we could see the same effect. So this is the scientific goal of trying to reproduce your results. And well, there's also the pathology professor, Dr. Koch, who had his postulates. He is proving that someone got ill from a certain bug. You had to introduce this, isolate it from the patient, introduce it into a healthy patient, and when this one got sick, you proved that this was the agent. So in fact, we do that now for these zinc finger transcription factors. And now what you did in this experiment was taking this zinc finger module, equipping it with VP16, so an activating domain, having also another construct where you had a repressing domain fused to it, and then introduce it into the same plants. At least you try to find plants, or you don't try to find, or you don't want to know, but at least you try to see what happens when you have a kind of battle in the plants where there's a competition of the zinc finger domain to each of its possible sites. And you might have the situation that, yeah, that for each gene there's a competition between activation and repression. So largely it's very unpredictable, I would say, what will happen.
Inside each bud is obviously gametes, and they generally self-fertilize before they even open or after they open. And so what we have is each flower, actually, each embryo has a competition inside of it. So the landing of the zinc fingers won't be the same in each embryo. It'll be different in each seed. Is that true?
Yeah, well, if you speak in genetic terms, you could say, well, the landing of you should discern, I think, discern the insertion of the transgene, so which is the gene which you introduce via agrobacterium, and you do this by introducing it into the you could say the germline of plants. Well, this gene construct has to integrate into the genome, so this can happen at any position. But the point is that once it is integrated, it starts to be expressed, and then you get this protein, you get this artificial transcription factor. And this will then start to find its target sites. But if you have introduced at the same time in the same tissue, the same ovary in this case, you have introduced another zinc finger construct, which has another action, then you will have in the developing embryo a kind of a battle between these two different transcription factors.
So each embryo will have a different result. Say if there's 1,000 possible places that either the activator or the repressor can land, each embryo will have a different ratio of repressors versus activators. So each seed will actually have a different result to some extent.
Yeah, it's very unpredictable because what will happen. So.
Yeah, I wanted to talk about the effects of reprogramming up and down at the same time. I think we kind of got there. I did want to get to this question. It was a little bit about demethylization. I'm not even sure exactly what demethylization is. But if we talk about epigenetics and deep time genes, the idea that there's a lot of genes that aren't expressed generally, but only are expressed during catastrophe. or during stress, intense stress. And I'm sure there are many in ourselves that if the atmosphere drastically changed, we might suddenly have a fallback plan that we don't usually express, like adrenaline. When you need to lift a car off of someone, suddenly you have the adrenaline. But what I know that we're getting close to canalization and atavistic things. So, and I know it's something that you like to talk about. And for me too, I like to talk about what we are. I guess I was really inspired by some of the things you said, that we're sort of not just our genome, but also the habit of our genome's expression. And a lot of it is just a habit. That's, I think, what you call canalization, where you're as an organism, you decide not to use everything you have, but to get in the habit of using more or less of what you have. Is that if you want to define canalization a little better than that, but I'm quite interested.
Yeah, well, if you look at the canalization in the sense that as you mean it, I think this term was coined by Waddington in the 1950s, maybe a little bit earlier even, and he didn't know about that much about genes and transcription, etc. But he just tried to formulate a theory where he said, well, why is an organism stable? Why is it as it is? Well, because you have to realize that you don't want cells to be completely erratic, at least in normal development, because then it's very unpredictable what they will become. So you want your liver cells to be a liver, and your heart cells to be heart cells, and your brain to be brain cells. You don't want them to change fate and development.
I don't want to live brain.
You wouldn't like to have that. So. And so he said, well, there are so many challenges in the environment, so somehow the organism needs to be very well buffered. So there needs to be some kind of buffering system in development that once you go along a certain path, that this path becomes more or less stable. So he made some kind of an idea that he called it an epigenetic landscape. You say, well, it's like rolling a pebble down a hill. And when it finds a little valley, then it goes into there, and when the valley becomes deeper and deeper, it can't get out. So that's the developing embryo. It finds its way, and by finding its way, it also gets stabilized. So its development is stabilized.
And these are like fate maps.
Fate
maps and things.
Once you are in a deep valley, you can't get out.
Right. Once you're differentiated, it's hard to de-differentiate.
Yeah, it becomes more and more difficult. So if you are in a shallow one, it's maybe still possible. If you receive some kind of trigger that you switch into another gully or valley, but if it's too late, it's nearly impossible.
But one of the things that you're trying to do is actually advance upon this buffer zone and sort of like stress the organism enough that it may even slowly roll out of the valley and over a mountain and into another form under duress or interrogation, but also finding its own underused genetic propensities. Like this is what atavistic genes are. I call them deep time genes because they're actually to some extent relatively unused until there's nothing else to use. Like it feels like everything's in disarray, but there's this. We haven't used this in a long time, but maybe it will help. I know the genome doesn't have this type of intention, but I gather your image of going up out of the valley, up over a mountain to the other side as a change in phenotypic regularity. It's sort of like is already a possibility in the body, but it needs a trigger.
But in fact, our body does these things because we have in our, I don't know how many different cell types there are in the human body. All of these cell types have more or less a stable expression pattern. So cells have acquired a stable state, which allows them to be different while having the same genome. When I was studying, I was even told by people that, to explain this phenomenon, they said, well, the cells have to lose their DNA, at least part of it, to explain this. Well, we now know that for a long time already that this is not true. The cells have the same DNA, but somehow they acquire a state where the gene expression pattern is stable, and this is then correlated with the cell identity. So this is a process which takes place gradually, and if we can influence this, well, we might. That's in fact what people try to do also with stem cell research. Stem cells can still go along all the different paths, but if they become more older, more gone through into different fates directions, then they get gradually less pluripotent. So they,
when they go from totipotent to pluripotent to differentiated down different pathways from bone marrow cells into white blood cells, etc. Is that, how do I say, is that methylation? Is that when certain genes get covered up and you only have a few genes that are more active than others?
yeah, if you talk, then you talk about epigenetics. So this is not the true genetic code, but this because that you can say the genetic code is what's written in a DNA. So these are the 4 different bases, well, letters A, G, T, C. Well, but the epigenetic code is something which is superimposed upon that. So we could say our genome is the hardware which is embedded in epigenetic regulation. And epigenetic regulation is then associated with the presence of proteins. These could be histone proteins or other proteins which might carry structural modifications, or even the DNA itself might have some modification, which is methylation of cytosine, for instance, which is one of the bases. It's a very common methylation. It still behaves like a C during replication and also during all processes, transcription, but it carries this mark. And you might say that methylation of DNA is correlated with this part of the genome being less active in terms of transcriptional activity. And there are also these protein modifications of the histones. There are also codes upon moieties attached to these histones, which also carry information either to enhance or repress the transcription of the genes, or that the whole, in the worst case, if you talk about inactivation, it might be that you could imagine that the whole stretch of DNA becomes really compact and like heterochromatin. So the thing is really condensed and very difficult for other factors to see the information which is there. Which the result is then finally that this gene, this area of the genome is no longer expressed.
This is what I call genes that have been seriously repressed in certain cell lines or in certain whole organisms, right? Mm-hmm. There may be a lot of, to be simplistic, our tail gene. I think our tail disappears at the same time our genitals are forming, so maybe it's being used. But I don't know about that, It's like...
Right, it could be, maybe it's a bit of speculation that you say, well, if some other type of gene expression pattern would be present, we should have kept our tail, for instance. I don't know whether we would be happy, but yeah. No, but it might be. You might. I don't know whether these genes are still there, but also for snakes, well, during evolution they have lost their legs. Mm-hmm. So maybe there's a possibility to grow them back if the genes are still there and you could reactivate them. If it's only a matter of activation again, rather than that they have disappeared or severely have been mutated, well, then it might be possible.
I like this. We had talked earlier, and one of the things that you said, I don't know if you can say it again, but it was just that after you do a whole genome interrogation of a plant, there's some chance that it might not even be completely defined as a plant anymore. And I think what you were alluding to was the idea that some genes from before plants existed might be atavistically contained and demethylated or upregulated, or in response to downregulating, come back into use, gene expression, gene action, become activated. Is it possible that some genes that have been in these plants since before plants existed might actually get turned on or differently regulated?
But you could say that everything which is still present in current genomes should have more or less a function. But we know now that this is not true, because there are large parts of DNA which you could call junk DNA. They will have a function, but so there's lots of stuff around of which we really don't know the precise function. So it might be that there is something there. Yeah, of course now with the sequencing of so many genomes and comparative genomics, well, there's maybe less room for a lot of speculation, but yeah, you could say if these genes are there, and of course you see the tree of life really reflected in all these genome sequences. So there are, of course, things we still share with the bacterial world, and maybe they have been adapted to have eukaryotic expression signals, but we share these same genes. Well, you never know what happens when you have an imbalance in this gene expression, at least an imbalance meaning that it is different from what's normally happening. So you don't know what happens. Well, I don't want to say that we could turn into bacteria. That would be complete nonsense. But certainly, yeah, you could have different patterns of gene expression, and if they will be stable and not toxic or lead to too much distortion, you might still generate a cell with this DNA, which is normally still present, and you might also generate still a multicellular structure. And yeah, depends what you then finally get. Maybe most of these things, if you do it, will be toxic. Right. So as we do it now with the genome interrogation, we introduce these transcription factors into plants in this case. We see plants appearing, but we also see that a lot of plants probably don't make it.
And they're infertile or...
Yeah, or even die maybe shortly after embryogenesis. Mm-hmm. So those which survive, and to me that is remarkable. There are of course very strange phenotypes, and I think we have looked at a few pictures. Well, you would still, I think a keen botanist would still know that it is something like a crucifer and it might be Arabidopsis, but for some of these plants it's very difficult to tell. At least for a layman, I think it's not really easy to say what it is. But also the surprising thing for me is that so many plants are still relatively normal. You don't see that much. Maybe you would see something when you would dive into it and do all the metabolomics and et cetera, whatever you can do. But so there seems to be also some kind of resistance in an organism to changes. So it's not so easy to change something. So this really confirms in my idea the epigenetic landscape, which is, as Weddington sees it, with these valleys where something needs to go through. So if you see a mutant phenotype, it's probably something which has entered another valley. But the mainstream plant behavior, the mainstream phenotype, is still very much very strong. It's very robust. So it's very difficult for a transcription factor to change it. Yeah. But we can, but I think. But it's the normal pattern is very well buffered, and that you see that also in notions in literature, that people talk about phenotypic or genomic buffering, and this is all to explain that there are many changes which have occurred in the genome, all kinds of mutations, but still you see very little effect of those. Phenotypes want it. So this is a paper about Arabidopsis where they knocked out all kinds of genes, but they see so few phenotypes.
Yeah, I know, because when I was at MIT, I worked near Nancy Hoffman's lab, and she was doing high throughput screening of zebrafish with something that caused point mutations like EMS. She just wanted every knockout possible, and people were similar to your research, a little bit like, why would you want to do that? They were working on one gene their whole career, and she wanted every knockout possible. She learned a lot about embryogenesis, even from the lethals, because if you can find out what got knocked out that made it lethal, you find a very early developmental gene. Mm-hmm. But I kind of like these high throughputs, and it's something I wanted to talk to you about, which has to do with, what's it called, combinatorics or permutations. Mm-hmm. For me, I can see you're using different lengths of landing pads, like 9 base pairs or 18 base pairs or things like this. But for me, I also see that you're doing something like all permutations possible with this zinc finger, or every zinc finger possible to be used. So all 9 base pair combinations. To me, that's permutative genetics. And it's also, I explained to you a little, for me, it's a little bit like Kabbalistic early algebraic, where you're trying to actually do all permutations to create life, to create like a golem or something like this, like to make your own life. But when it comes to permutative, in my mind, if you're working with body plans or phenotypes, you're working with cell fates. And it seems to be that you're working on an attempt to express as many variants as possible, like all potential variants in an organism, like even to make new kinds of trees that have never existed, but also just to see what an organism can be. Maybe it gets back to that Spinoza thing, like we don't even know what a body can do. Let's start with that. So we're actually interrogating a body through not just seeing what one gene changes, but actually seeing what it means to sort of tweak the genome knobs in every direction. And I wonder, with that kind of, to me it's a sort of aesthetic. It's kind of like a conceptual aesthetic, which is to say, let's see the possibilities before we make up our mind. So it's not trying just to engineer, but it's actually trying to do a high throughput mutagenesis. And that seems like some of what you're doing, and I just wonder what you think of that as a way of describing it, or whether you would describe it differently.
It's a way of finding mutants, but it is it's transgenic in this sense that the thing that you introduce is now what we do is a gene which encodes an artificial transcription factor. But still, you might think could be possible that you would introduce only the protein. So then you don't change the genome contents of the cell, but you introduce the protein. And if you could introduce this at a sufficient level, this might start to influence gene expressions. It might start to influence the epigenetic landscape, if you like, and it might start to influence further development. So this is just proving that something can be done by having an artificial gene construct. It's not necessarily forever linked to making a genetically modified plant in my case. So you might say, you might trigger this behavior, which you hope that you like it, in case the plant grows better or makes better fruits, whatever. If you could introduce this trait by, spraying it with proteins, then
the protein therapy.
Then it would be protein therapy.
And then it doesn't actually. Then it's not a genetically modified organism. I guess it would be protein modified organism, but we would call it or protein. It's just spray onto the flowers and then collect seeds, right? But you could spray the protein on.
Yeah, you could try, but the difficulty is, of course, to get sufficient amounts into the cells of interest. But But the main thing is, I think that with this technology, it's not that you. Of course, it is a mutation because you introduce a piece of DNA, but that's not the prerequisite, really, because you might think of the possibilities introducing just a protein, and then you would train maybe a genome just to behave in a certain direction, which will finally generate another phenotype. So that's probably a lot of nonsense I'm saying now.
No,
But,
that's pretty good. I mean, what I was asking, actually, part of what I'm saying is that also that you could. I know that you're going on a fishing expedition to see if you can improve or optimize, crops. It's sort of like one of the reasons that you do your research. I'm sure you do pure research too, and I privilege pure research because I like the fishing expedition itself. In other words, when I say you're looking for phenotypes that are useful for industry or useful for, how do I say, maybe even pharmaceutical production, et cetera, but it's, for me, I'm interested in, by doing whole genome interrogation or whole genome reprogramming, you're actually expressing a wide an array of different bodies from a single genome that you possibly can first. Yeah. And then you sort of sort them after that. That I'm sort of fascinated with mutagenesis, all like you can hear, like in and of itself, without a purpose for. I don't need to make better oranges. Personally, I'm an artist. I like it. I might make a plaid orange, but I mean, mutagenesis itself is a creative force. Like gamma rays are creative. They create difference. We would still be single cellular organisms without mutagenesis. So I don't consider it to be like the horrible thing. I think it's a river of change that flows through all time. This is evolution, and it's not always pretty, but it always changes. So I like delta. It's a preference. But I had talked to you about actually you yourself with your mutagenesis experiments are, as a person, a mutagenic event on Earth. Like you are a mutagen because you're sort of stirring the pot more than most. And I don't know. If you cause more mutation than the average person, are you Bert a mutagen yourself?
Yes, possibly, yeah, but of course our safety officers don't want us to leave them out in the open. But well, you could say, yeah, we contribute to evolution. Yeah, we could do so, but we keep them inside, of course. But yeah, we don't do that much more than anyone else. I think if you consider mutagenesis and evolution, I think everyone is part of this. If you can have a holistic view about this, well, everything is connected, and you could also have a Spinozistic viewpoint. Say, well, what's happening is nature, which is God, and we are part of this. So we are connected with everything, and then yeah. So if you are a plant biologist, a molecular biologist, it's maybe rather straightforward to see this, but if you have come over from the States, you have a lot of fumes from your airplane. Well, this might also cause organisms to mutate. So also your behavior contributes to further development and evolution.
So maybe I need not just, as an American flies a lot, maybe I need to not just think about offsetting my carbon credits, but offsetting my global diversity genomic credits with every flight, yeah. What do you think of this idea of this bipolar flower, this bipolar plant? And I'm going to amend this. We have 6 zinc fingers, but only 2 constructs. One for upregulation, one for downregulation at 9 base pairs. But they're competing in these buds right now. There are competitions that are starting in every embryo. The results of the competition will be a little bit different because these constructs are pulling the gene expression up and down in opposite directions at the same landing points. For me, I call this bipolar referencing like the mental condition, but also because a lot of artists have some of these conditions and I like them. I still like them. And so if I make a bipolar plant, it's not out of like disregard, but actually it's a little bit of love for the crazy people I know. And if we have this multiple hundreds, maybe thousands of genes with altered expression patterns, it repaints possibly phenotype or anatomy, it repaints possibly metabolism. It even could repaint the scalable product, right? Like how much we can get from them. But what do you think about it as merely as a sculpture? So like these plants just being altered, their gene expression being altered with up and down regulation in the name of conceptual art or sculpture, or actually calling this plant doctored in the name of art, not doctored in the name of knowledge production or science?
Well, I think with a sculpture, I think of something very static. I think a sculpture, it's there. It's just made of stone, maybe, and I see that very static. But and these plants, I think, what is happening there? Suppose that you indeed get a situation that you have both of these constructs present, you will have a very dynamic situation. And in each different plant, well, suppose that all these different plants still have the same. The 2 genes, well, the level of these genes expression will be different because each of these genes has landed in a different position in the genome. So you really don't know what to expect. So that way, it's very different from, I think, from a scientific experiment. This is something it's difficult to repeat because you don't know what happens, and even if you would reintroduce these 2 constructs again, you will have a very difficult, very hard time just to point out this one which was interesting for you. So each of these plants is different. I don't know for art. Well, if I don't know if is art reproducible.
We try not. There are some conceptual minimalist artists that pride themselves on reproducible art. It's usually nonsense, but reproducible nonsense. But for the most part, we try towards this sort of modernist avant-garde, if you're that old-fashioned, I guess, but to be novel or to be original all the time. It's almost the opposite of science, where it's as unrepeatable as possible. We also find that scientists actually come up with more non-repeatable things than artists, and artists are quite predictable. So that's a question.
But might still be interesting because this, well, one of these constructs with the activator, well, we find it produces bigger plants. So yeah, not all the genes that are disturbed in their expression pattern are probably required for this. So it might be that if you have a situation where you also have a repressor present, that you might find a better balance of those factors which are crucial for triggering the true phenotype of interest, which is a plant with bigger leaves. So it might be that this project contributes to some understanding, but it's very difficult to reproduce it in lab conditions. I think to reproduce this particular plant is by this strategy. So that's why normally you wouldn't do it, but yeah, maybe now it's done now, and maybe there is still some extra benefit from this project. Let's hope so.
Well, here's the thing. You know I'm trying to avoid extra benefit as far as like, I don't want this to be useful at all. But this was the thing that I liked about you, Bert, is you actually got this joke. Your grad student, right, Niels, was sitting there, and after a while I said, I really don't want it to be repeatable. I don't want it to be able to be backward. I don't want to go backwards with it. And in particular, I don't want this to end up being applied science. I actually wanted to stay unapplicable. And you actually, I'm not sure exactly all the ideas you came up with. One was the idea that we would use the same marker for all the constructs, and we didn't really. But the other idea was that we would do what we did, which at first I was like, what? The idea that we would use an up regulator and a down regulator on the same zinc finger. I thought you were just being cheap. And then I realized, no, he's saying this will make it harder to be repeatable. It's make it harder to even figure out what we did because of this competition for the same area. And so how is it that someone who spends most of their time trying to make good, solid, repeatable science can still have a sense of humor about making something that's actually completely just an artistic application of biotechnology? Yeah.
Well, maybe I'm easily persuaded to follow another kind of thought. I don't know, but yeah. But somewhere hidden in everyone, I think there is a playful person. So maybe that's the reason. I don't know. Yeah.
Yes. Okay. I know that if I mention vitality, and I also attenuated that, or the random walk of life, but we've talked about vital forces, like what is the force of life? And it's not something that, I'm not a very religious person, but I actually think that life has a force maybe beyond just chemistry and physics. I don't know, I guess we talk about secular vitalism these days. That there is something to the biological sciences that's maybe fundamentally different, or at least something worth looking into about life's way of building. Mm-hmm. That's like maybe because it's recursive, maybe because it maybe you want to talk about your time frames. To me, what I wanted to talk about was I'm trying to make an enigma because I think that life is not just searching for answers. It's also enjoying the level of questions that it provides for us, right? And I wonder maybe if there is a place where art and science almost meet here, which has to do with the fact that no matter how much we try to comprehend, we're always in a world of artifacts and enigma. You know, that I know from talking to you that the further you go into science, the more you realize how little people know, you know, like how much mystery there is. So what would you say about just the idea of biology and mystery, or biology and random vitality?
Yeah, it's a bit of a difficult question. I think it's life in general is still a mystery to all of us. I think there's still no simple definition. For instance, it's very difficult to define life. Just in one sentence, it's I think impossible. So somehow the life as we know it, it's somehow inherited from some piece of space where you had a reproducible presence of low entropy, let's say, so that means high order. So this high ordered state was inherited by systems. Well, these got themselves equipped with nucleic acids, and well, this helped to propagate this state. But all of us, we still originate from maybe this event somewhere where a piece of order was, no, not maybe yeah, you could say created, but not necessarily. Well, it happened to be there, and we just are still living proof that this thing existed. So this original cell, which finally came forth from this order, well, this is our progenitor. So it has brought us into existence. Well, this could be maybe a very dull way of explaining life if you don't want to evolve all kind of theology of purpose, but it's there, this state where this order happened to be there, and yeah, we are still the remnants, and we are still the carriers of this order, and our destiny seems to be to pass it on. And if we have fun at the same time, I think that would be great.
Yeah. So, I would ask one more question, and probably this is, I'm looking at these extra ones, and really all I want to talk to you about right now, because we're doing this partially for biosolar cells, would have to do with, how do I say, maybe what you are allowed to talk about in terms of increasing photosynthesis. And in this question, it's a little bit plus or minus. We had been talking about this, making solar zebrafish with hoob, and eventually having transparent people with algae all over them that would then live off of sun energy and not need to eat. And this is a utopia. And some of this I said, well, maybe this is dystopia. You know, like maybe no one would want to work if it was a sunny day. Any day the sun came out, people would be like, why would I work? I have food, I have rent. I'm like thermodynamically self-sufficient. Leave me alone. And how would that help the economy? I don't see it helping the economy at all. But I also want to base this in the serious. You're also working on some increases of photosynthesis and what you're allowed to talk about. If you're using zinc fingers, if you're interested in sort of saying how it actually might help the economy, the ecology, the world, or what ease of human life, I'd give you that. This one's more for FOM and the biosolar cells guys. So,
well, what we do, you could see as well this genome reprogramming, because well, people you can have an alternative approach, which is that you study all the kind of natural accessions or variations or races or whatever of a plant species in this case. But people have done so for Arabidopsis. They have collected, yeah, you can find it on the internet, hundreds of different what they call ecotypes. And you can study all their properties and you see variations. It's all within one species. But each of these accessions, these races, they carry, or lines, they carry many, many mutations. But yeah, so you might learn something from that. You might find one of these accessions to be performing better under conditions which are of interest. So you might say, okay, now we get a higher CO2 levels in the atmosphere, so plants might profit from this. It might be drier or some places, or wetter, or there might be more salt, whatever. You might try to find natural plant lines which might perform better. But still then, if you want to get to know why those plants do better, you enter into a very difficult field, because then you have to prove that certain genes or expression patterns of genes or certain types of point mutations are crucial for this novel phenotype. So this is also a lot of work, but we do this in a kind of other way. We say, well, you have a given genotype. There are no differences in the population. It's one line of Arabidopsis, which is homogeneous. And we try to see what this plant line can do with its own genome. So we let this, we kick this genome, this gene expression pattern with artificial transcription factors. And then we can see, can this plant do something which is useful? And if it can do so, we can learn from it, because we can find out which genes are under influence of this transcription factor. It's difficult, but it's possible. And it's an alternative approach to studying natural variation, which in fact means mutations. So now we study, you could say, epigenetic modifications. rather than lots of point mutations. Yeah. So it's a complementary effort, I think, compared to studying all the different types of plants you can find, all the different lines. But now we start with one genotype.
And with the Arabidopsis, what about the idea of increasing photosynthesis? So actually increasing, do you think it's enough, actually, even though it's hard to begin with, to do it, do you think it's enough to increase a plant's photosynthetic metabolism? In other words, just to be able to more efficiently digest solar energy or more efficiently metabolize solar energy. What could it possibly do, or would it be enough for for positive utilization? You know.
Yeah, you can't maybe expect that you will work miracles, but I think every person in agriculture will agree that if you could improve yield by 5 or 10%, this could be very useful. Suppose that your crop growth facilities are optimal, then another increase of 5 or 10% is great. If you are a very careless farmer, of course you can already improve a lot by being more careful. But if your crop already performs at its top, and if you can still improve this top by making photosynthesis better, or making certain properties of more interest to people, then this is very welcome. And I'm not personally getting any money out of this, so I don't care. But just out of curiosity, I would be very interested to know. And I think also it might help mankind if we can really improve important traits in crops. And if we can do this in an organized manner, and if we know what we are doing. So if we know how to address complicated issues by a certain type of mutagenesis, and maybe this might be this zinc finger-based genome interrogation. Well, this might be a tool to improve performance of crops, but it can also be used to improve crops which are already performing very well. Because you could take a plant line which is the favorite of farmers and all over the world, for instance, or which is already known to perform very well. And then,
corn, wheat.
Yeah, but you could maybe then still see whether you have room for further improvement. Well, if you start from scratch, from making mutations, you have to assemble everything again in an optimal crop. Well, this is also very complicated, and it takes a lot of time. But we can start in principle with a crop of high quality, and we can, by using this genome interrogation, we can try to see whether it could perform better, given that you kick the system and that you run the development slightly different. Or that you have environmental conditions which might trigger the plant to behave differently, because now it can behave differently, because there is a trigger which keeps it out of these valleys which Waddington would otherwise predict. So you could maybe change the development and then end up with a better plant under given certain circumstances. So apart from scientific curiosity, I think we could also benefit, but at least I hope so.